Hi everybody, this is David Lally with the Partnership for Research and Education in Plants, and today I'm going to show you how to make observations on your Arabidopsis plants. Here I have 4 pots of Arabidopsis plants, a pot of wild-type plants and a pot of mutant plants that I've grown under control conditions, and a pot of wild-type plants and a pot of mutant plants that I've grown under experimental conditions. In the control conditions, I've done my best to grow the plants under the best conditions that I can provide for them. And under the experimental conditions, I've altered one of the factors in their environment. In the case of this experiment, I have grown the experimental wild-type and mutant plants under high light conditions. Now, on to the observations. When we conduct experiments, we change something and then try to measure the effect of that change. Some people call the thing that you change the independent variable. Well, in this experiment, there are 2 independent variables. The genetic change in the mutant plants, in most cases a single gene has been disabled or knocked out. And the environmental change, that's the change that I made in the plant's environment, in my case, high light. We're going to examine the effects of each independent variable separately, and then we're going to examine the effects of the 2 independent variables together. Let's look at the effects of the genetic change first. Let's move those experimental plants out of the way. And I'm going to put the wild-type control and the mutant control pots side by side like this and see if I can discover anything different about the mutant plants compared to the wild-type plants. When I do this, I'm trying to answer the question, what are the effects of disabling the gene? Remember, I haven't changed the environment in this group. I just want to see the effects of the genetic change by itself. Record your observations and try to collect data that provides the best evidence for your claims. I recommend putting the 2 pots side by side like this and taking a picture of them, and then writing your observations right next to the photo. Now let's look at the effects of the environmental change by itself. To do that, I'm going to put the wild-type control pot right next to the wild-type experimental pot. When I do this, I'm trying to answer the question, what are the effects of changing the environment? Remember, I haven't changed the genes in these groups. Both pots contain wild-type plants. I just want to see the effects of the environmental change by itself. Again, record your observations and try to collect the data that provides the best evidence for your claims. And again, put the 2 pots side by side like this and take a photo of them and write your observations right next to the photo. Finally, I want to examine the effects of the 2 independent variables together. This mutant experimental pot has experienced both the genetic change and the environmental change. To examine the effect of the combined variables, I put the pots back into the 2x2 arrangement that I started with. Here are the wild-type and mutant plants that I've grown under control conditions. Remember, these are the ones I compared to examine the effects of the genetic change. And here are my 2 wild-type pots, the ones I compared to examine the effects of the environmental change. I'm going to compare the mutant plants grown under experimental conditions to the first 2 comparisons that I made so that I can answer this question. Does the genetic change affect the mutant plant's response to the environmental change? Let me give you an example. Okay, so here is my mutant experimental pot, and I'm going to look at these other 2 comparisons that I've made. First, what's the effect of the genetic change? And I can see in the mutant here that it's not doing as well as the wild type. I know that because they look smaller. That's something that I can confirm with a quantitative measurement by measuring the distance across the plant itself. I can also see that there's some curling up of the new leaves that are emerging from the center of the rosette. That isn't happening over here in the wild type plant. And the texture of the leaf is a little bit wavy over here too as well. That isn't happening on the wild type plant. So these are effects that are specific to this gene being disabled. Now if I look over here at the 2 wild type plants, here's my experimental and here's my control. I can see under higher light conditions that the wild-type plants look much more robust. They're larger, darker green. I can see the flowering bolts emerging from the center of the rosette. And I know that these plants are well on their way towards their reproductive phase. So now I've seen what's the effect of changing the environment, what's the effect of disabling the gene. I can look over at my experimental mutant pot and I can ask, is that response different to the high light conditions? And it certainly does look different. They're a lot worse off than the mutants under control conditions were. They don't seem to be thriving as well, and I can see that for many reasons. Over here I can see that on the surface of this leaf, not only does it have that wavy pattern and is it smaller, but it has a dead spot, a brown lesion on the leaf that has appeared. If I look over here and say, well, is that just the amount of high light? I can see that there are no brown spots on the wild-type plants. They all look very healthy. And I can see that none of these are bolting like the wild-type are. In fact, they seem to be really struggling to get out any new leaves from the center of the rosette. What I can conclude from that final observation is that the mutant experimental pots are growing much worse than their mutant control counterpart. Now, if you look at the wild type, they're growing better, the wild type experimental, than the wild type control. The mutant is growing worse under high light conditions. So that's a difference in the way the mutant has responded to my experimental conditions. And that's the kind of thing I'm looking for to answer my last question. Is the response different in the mutant plants than the wild type plants? So I hope that's helped, and good luck with making observations. Check out the collecting data video.